Video is about mutations. We're going to be covering what are mutations, the types of mutations, and the causes of mutations. So to begin, what are mutations? Now we know that traits are inherited from our parents. However, some traits are not inherited from parents. These traits can be a result of a change in DNA, or in other words, a mutation. How can we define a mutation? A mutation is any change in the DNA of a gene or a chromosome. So it is a change in the organism's DNA. For example, instead of the base sequence adenine, adenine, guanine, the DNA sequence may accidentally have adenine, cytosine, guanine. Now this change can result in the production of a different protein that may code for a different trait, and this trait might differ from what it would normally be. Now, many types of mutations can occur. Typically, mutations that affect a single gene happen during replication, whereas mutations that affect a group of genes or an entire chromosome happen during meiosis. So we will begin with mutations that affect a single gene, which are also known as gene mutations. There are two types: point mutations and frameshift mutations. Point mutations occur when one nucleotide is substituted for another. That is, an incorrect nucleotide is put in the place of the correct nucleotide. An example of a disease that is caused by point mutations is the sickle cell disease. The second type of gene mutations are frameshift mutations. This involves the insertion or deletion of a nucleotide in the DNA sequence. An example of a disease caused by frameshift mutation is cystic fibrosis, which is the overproduction of thick, sticky mucus. So a question may be, explain which mutation you think would have the greatest effect. This would be the frameshift mutation. Why? Because they shift the entire sequence following them by one or more nucleotides. Next is chromosomal mutations. The first type of chromosomal mutations is gene duplication. Now recall during meiosis, specifically prophase, homologous chromosomes, which are chromosomes that have the same order of genes, homologous chromosomes exchange DNA segments through crossing over. However, if the chromosomes do not align with each other, these segments may be different in size. As a result, one chromosome may have two copies of a gene or genes, and the other chromosome may have no copy of the gene or genes. The second type of chromosomal mutations is gene translocation. In translocation, a piece of one chromosome moves to a non-homologous chromosome. Translocation is often reciprocal, which means that the two homologous chromosomes exchange segments with each other. Now, translocation can result in a mutation that may break up a gene, which could make the gene no longer work, or it could make a hybrid gene that would have a totally new function, or it may come under the control of a new set of promoters so that the genes would be more or less active than usual. What causes mutations? Mutations just may be caused by replication errors. So we know that the DNA polymerase has a built-in proofreading function. However, a small number of replication errors are not fixed by the DNA polymerase, so these mutations will build up over time and eventually affect how the cell works. Mutations can also be caused by mutagens. Mutagens are agents in the environment that can change DNA. How do mutagens change DNA? They speed up the rate of replication errors and in some cases even break DNA strands. Some mutagens can occur naturally, such as UV ultraviolet rays and sunshine, and other mutagens may just be industrial chemicals. So that is all for today's video.